Pigmentary glaucoma is the progression of pigment displacement syndrome, a condition characterized by the release of pigments into the anterior cavity of the eye, particularly within the trabecular meshwork cells shown here. This is believed to be caused by friction between the posterior iris surface and the anterior zonular bundles that causes liberation of pigment granules. This condition is genetically caused by multiple mutations showing patterns of inheritance that is debated as either familiar aggregation or autosomal dominant with incomplete penetrance. There has been gene localization of a susceptibility gene to the Q arm of the 7th chromosome, but no specific gene has yet been found. Progression of PDS to pigmentary glaucoma is typically seen in white males between the ages of 30 and 40. This advanced condition is observed through higher intraocular pressure than PDS patients, as well as pigment showers and increased pigment displacement caused by physical exercise and quick eye movements. However, this disease is frequently asymptomatic, which can be troublesome as current intervention treatments for PG decrease in success rate the longer it progresses. There is currently little understood about the causes of the progression of pigmentary glaucoma from pigment displacement syndrome. One of the hurdles for pigmentary glaucoma research is the limitations of previous model organisms used. Rats, a very common research animal, have eyes that are too small to be informative for pigmentary glaucoma, and they have very little amounts of TM tissue for making ex vivo cultures, an important source of information for pigmentary glaucoma. Apes, on the other hand, are very useful as they can develop intraocular pressure, but are quite costly to use, making it difficult to have a study with enough animals to be relevant. Porcine or pig have a lot of advantages as a model organism. They are readily available samples from abattoirs, especially eye samples for pigmentary glaucoma. They have high quality tissue with low culture time of about 2 hours. There's consistency amongst litter, so there is less variation between sample to sample. And there are similarities between human and porcine outflow tracts in the eye, making testing for pigmentary glaucoma with pigs informative. Pigs also do not experience ocular hypertension in nature or through medical intervention, reducing confounding factors such as procedures performed for sample preparation. This group has developed a porcine eye model of pigmentary glaucoma that replicates clinical features seen in human patients, such as higher concentrations of pigment granules in aqueous humor and inside TM cells. Pigment granules were not cytotoxic to cells, with levels of apoptosis being the same between the control and the treatment. Pigment introduction increased with intraocular pressure, and affected cells had greater variation than the controls. Pigment in TM cells of pigment dispersal eyes showed activation of lysosomes and phagosomes within both ex vivo and in vitro samples. These pigments were being swallowed and hydrolyzed by secondary lysosomes. Pigment accumulation decreased phagocytosis in TM cells. Gene expression microarray of pigmentary glaucoma model showed differences in cellular movement, cell adhesion, and endocytosis and phagocytosis signaling pathways. This porcine model is the first ex vivo model of pigmentary glaucoma to date in any species. Results suggest ocular hypertension in pigmentary glaucoma is the result of actin stress fibers, reduced phagocytosis, and cell matrix adhesion. Pathway analysis revealed that the activation of the Rho-A pathway played a central role in TM actin cytoskeleton, tight junction formation, phagocytosis, and cell motility pathways. These highlighted pathways could possibly lead to targets to treat pigmentary glaucoma.